In this session today, we'll look at metastasis, the final phase in cancer development. Metastasis is defined as the spread of cancer cells from the primary site to distant locations in the body or the secondary sites. It accounts for more than 90% of the cell deaths that we get in cancer. And despite this, it still remains one of the most poorly understood processes. Most tumors start off being benign, and a vast majority of tumors are benign. However, some of them undergo the next phase, which is the malignant or the metastatic phase. And this is when tumors break through the basement membrane and go through the barrier into the blood and the lymphatic systems. And using this process, they pass to different locations. The basic process of metastasis can be divided into several phases. The first phase is local invasion, and this involves the tumor cells breaking through the basement membrane and out into the blood. The second step is intravasation. What this means is that this cancer cell passes in between the endothelial cells. Endothelial cells are a type of cell that lines the blood vessels, the smallest one, which is the capillaries. So they squeeze through the endothelial cells and move into the bloodstream. And this is where they interact with the various components of the blood. The next step is circulation. So this is the phase when the cancer cells pass through the circulatory system into the various, through the heart, and then to the various organs such as the lungs or the bone marrow or the brain. And from there, they go through the phase of extravasation. And this is the phase where the tumor cells now move out of the bloodstream into the surrounding tissue. And this is a reversal of the intravasation phase, where now the cells squeeze through the endothelial cells to get past them and back into the tissue. And after this, you get the formation of a microtumor. So once these tumor cells get into this secondary location, they may start to grow and start to divide. And again, the same process of angiogenesis, as you saw earlier, can begin, where it forms a small in situ tumor, and then it triggers angiogenesis to get bigger and form a larger secondary tumor. Now, one thing to bear in mind is that metastasis is a highly inefficient process. What that means is a vast majority of cells don't actually make it from the first site to a second site. Usually, most tumors stay in a dormant stage. That means they are located in the primary tumor. But once they go into the secondary phase, when they go into a secondary site, again, they may stay first as single dormant cancer cells, so they're not actually forming an in situ tumor, or they might form a microtumor. So again, it's a small lump. Or the last stage would be the final stage when they switch on the angiogenic trigger, and this enables the tumor to grow. Now, this is quite a complicated and fascinating process by itself, how tumors can move from one location to another. And one analogy I can use is something like shapeshifters. What tumor cells do is they change their morphology, not just the shape, but also the sort of genes that are switched on and genes that are switched off. So they turn into a different cell type to enable them to move and colonize a distant location. And in order to do this, a tumor cell should have two basic properties. The first one is an ability to invade another tissue, and the second one, its ability to survive in an anchorage independent manner. What I mean by that is that a cancer cell does not necessarily need to stick to a substrate or something to enable it to stick on and grow. What invasive means is a remarkable process called EMT, or epithelial mesenchymal transition. As I said to you before, cells will change the way they behave. And this change in behavior is driven by this EMT. And this typically would happen in various phases. For example, in this phase, when the tumor cells have to migrate or squeeze through the endothelial cells into the blood, the intravasation phase, what this enables it to do is break away from the mass of tumors and start living independently, individually in the blood. But it needs to be able to move. The second phase of EMT is usually at the extravasation phase, and this is again where a tumor cell has to physically move through the endothelial cells to get back into the tissue. So again, this is an ability of the cell to change its behavior from growing using a substrate to actually physically moving and invading another tissue. And here's the remarkable thing. Tumors morph again. Once they have colonized a secondary site, it undergoes a second round of change, the mesenchymal epithelial transition, where the tumor cells now go back to being able to grow and divide using a substratum. So they lose the ability to move and acquire the ability to divide and grow again. And this epithelial mesenchymal transition, I'll give you an example here using a protein called E-cadherin. Now, E-cadherin is typically found on the membrane of the cell. So it sticks like a little protein on the surface of the cell. And usually it has another protein called beta-catenin, which is on the inside of the cell. And in order to undergo this epithelial mesenchymal transition, what a cancer cell does is it reduces the amount of E-cadherin, so its ability to stick reduces. Instead, it increases the expression of a protein called integrin. Integrin is a protein, again, that sticks on the surface of a cell. It acts like the Velcro of the cell, and it enables cells to move. So cancer cells will now overexpress this protein called integrin and lose this protein called cadherin, which enables it to stick. And therefore, it changes the behavior of a cancer cell from a static cell to a cell which is able to physically move across to different locations. So this process of epithelial mesenchymal transition, I can give you an example of two key proteins that are changed in a cancer cell. One example is a protein called E-cadherin. Now, E-cadherin is a protein that sticks on the surface of the membrane of the cancer cell and enables it to stick to another cell. So it enables joining or sticking together of two cells. What cancer cells do when it has to undergo EMT is they reduce the expression of E-cadherin, and as a result, these two cells cannot stick very well. Instead, it increases the expression of another protein called integrin. Integrins are also membrane-bound proteins, and they act like Velcro. So they can allow this cancer cell to physically move across to a different location. So as a result of this change in expression, less of E-cadherin and more of integrin, a cancer cell can move. And this enables this EMT transition. The second phase of anchorage-independent survival. Now this is an important point to bear in mind, that cancer cells actually, a lot of them die when they are in the blood circulation, because the blood is a very harsh environment. The change in pH, etc., actually targets these cancer cells for destruction, or they kill themselves. Again, because of the acquisition of unique properties that enable these cells to not stick to its neighbor and actually be able to move to a different environment, allows them to physically move across to a different location. In the case of benign tumors, tumors that don't grow, these genes for metastasis are switched off. For example, is this intestinal polyp, where you can see clearly that usually it's only a benign tumor. It's just an increase in size. We've had a look now at how metastasis occurs, and in the next session we shall summarize some of the key hallmarks and also talk a little bit about the cancer stem cell and some other key stages in cancer development.